This case study reveals how our multi-omics research factory, Morph, was used by researchers from the IB Catalyst-funded Detox project to identify engineering targets to increase styrene production in E. coli. In this fed batch fermentation, a phenylalanine overproducing E. coli strain was further engineered with a two-step process to convert the accumulated phenylalanine into cinnamate and then the cinnamate to styrene. Multi-omics analysis of the styrene-producing strain are compared to a non-producing control strain expressing inactive enzymes to reveal the response to product toxicity during production, as opposed to the conventional exogenous chemical addition in a strategy termed multi-omic-based production strain improvement, or MOBSI. By evaluating producing strains in downscale bioprocessors rather than flask cultures with external additions of toxins, MOBSI can be used to identify engineering opportunities relevant to an industrial fermentation. Fermentation data shows that the production strain and the external addition control both stop growing in response to styrene accumulation. Samples were taken at each of these time points and analysed by transcriptomics, proteomics, metabolomics, and lipidomics. Morph was designed to display all of this data through an easy web-based interface so that correlations between different datasets can be identified with ease. The transcriptomics and proteomics are displayed in volcano plots and heat maps. The MOBSI approach considers responses within as well as across fermentations. The fold change and p-value thresholds can be adjusted, and Morph gives a list of genes above those thresholds. The ability to display different types of omic data together enables identification of positive and negative correlations. For example, CPXP was identified as a gene of interest based on its gene and protein expression. 4 categories of genes for further evaluation were determined. Class 1 includes genes that were significantly regulated and positively correlated in both transcriptome and proteome in response to styrene, like this one. Class 2 includes genes that exhibit significant differential transcriptional regulation in response to styrene, but the corresponding proteins were undetected in the proteomic datasets, such as AAEA. Class 3 includes proteins exhibiting differential abundance in response to styrene, but no significant change in transcription of the corresponding genes. Class 4, genes or proteins that did not exhibit differential regulation, but potentially contributed to changes in regulation, membrane lipid composition, or metabolite concentrations. The lipidomic data indicated an increase in unsaturated lipids, and by implication, increased membrane fluidity and permeability, suggesting that genes involved in the synthesis of unsaturated fatty acids might affect styrene productivity. In total, 23 candidate genes were screened for improved styrene production by deleting and overexpressing each gene in the production and control strain and testing them in shake flask cultures. The best producing strains were identified and assessed further for styrene production in fed batch fermentations. A re-engineered strain that combined the deletion of AAEA with overexpression of CPXP outperformed the starting strain for both the maximum specific cumulative rate of styrene production and the total amount of styrene produced, illustrating the power of Morph MOBSI to enhance the performance of cell factories. For more details of this research, visit our website morph-db.org. To find out how you can get more from your data, please get in touch.